In our analysis of 1,771 CSIR NET previous year questions, one unit stood far above all others in question count: Structure and Function of Biomolecules, with over 11,000 questions across all exam years. This is the single most important unit for CSIR NET Life Science preparation. This video applies the reasoning approach from that analysis to 5 carefully selected questions from this unit. Each question teaches a reasoning pattern, a way of thinking that you can apply to any biomolecule question you encounter, even one you have never seen before. The 5 questions span 3 major themes: protein biochemistry, lipid structure, and bioenergetics. You do not need to solve thousands of random questions. 5 well-chosen questions, understood analytically, will give you frameworks that apply to hundreds of similar problems. Let us begin. Question 1. Chaperone proteins Hsp70 and GroEL assist protein folding by: Option A, binding to exposed hydrophobic regions of unfolded polypeptides to prevent aggregation and facilitating correct folding. Option B, providing the structural template for the final protein shape. Option C, catalyzing disulfide bond formation between cysteine residues. Option D, synthesizing the protein from its amino acid sequence. Pause here. What is the fundamental driving force that makes newly synthesized proteins prone to aggregation? Identify that force, and the correct answer becomes clear. The correct answer is option A. The key principle is the hydrophobic effect. When a protein unfolds, its hydrophobic amino acid side chains, which are normally buried inside the protein core, become exposed to water. Hydrophobic exposure in an aqueous environment is thermodynamically unfavorable. If many unfolded proteins are present simultaneously in the crowded cellular environment, these exposed hydrophobic patches will attract each other and stick together, causing aggregation rather than correct folding. Hsp70 recognizes these exposed patches and binds to them, shielding them from each other and giving the polypeptide time to fold correctly. GroEL goes further by providing an enclosed, water-filled cavity in which a single protein can fold in isolation, completely shielded from surrounding molecules. Neither chaperone provides a folding template. The information for the correct structure is entirely encoded in the amino acid sequence itself. Option C is wrong because disulfide bond formation is catalyzed by protein disulfide isomerase in the oxidizing environment of the endoplasmic reticulum, not by cytoplasmic chaperones. The core reasoning framework: identify the hydrophobic surfaces exposed during unfolding, and ask what molecular mechanism shields them from causing aggregation. Question 2. The activation energy of a chemical reaction is reduced by an enzyme by: Option A, providing an alternative reaction pathway with a lower energy transition state. Option B, changing the equilibrium constant of the reaction. Option C, adding heat to the system. Option D, increasing the substrate concentration. Pause before reading on. Identify which of these options addresses the kinetics of the reaction, and which ones would alter the thermodynamics. Enzymes affect one but not the other. The correct answer is option A. This question is built around one of the most important conceptual distinctions in biochemistry: the separation of thermodynamics from kinetics, the equilibrium constant of a reaction, and therefore its standard free energy change, is determined solely by the structures of the reactants and products. It is a fixed thermodynamic property. Enzymes cannot change it. What enzymes do change is the kinetics, the rate at which the reaction reaches equilibrium. They do this by providing an alternative reaction pathway in which the transition state is stabilized at a lower energy level. The enzyme binds the transition state more tightly than it binds either the substrate or the product, thereby reducing the energy barrier. Option B is the classic trap. Many students believe enzymes somehow make reactions more thermodynamically favorable. They do not. The equilibrium constant is unchanged. The reaction simply reaches equilibrium faster. Option C is wrong because adding heat would increase the reaction rate by increasing molecular kinetic energy. But this is not how enzymes work. And heat would denature the enzyme itself. Option D is wrong because increasing substrate concentration affects the rate of the reaction but not the activation energy itself. Framework. Always separate thermodynamics from kinetics. Enzymes are kinetic catalysts, not thermodynamic modulators. Question 3. Both sphingomyelin and phosphoglycerides are phospholipids. Which one of the following statements is not correct? Option A. One has a fatty acid tail attached via an amide bond while the other uses an ester bond. Option B. The hydrophilicity of both is dependent on their head groups. Option C. Only one of them may contain a carbon-nitrogen bond. Option D. Both may have choline as a head group. This is a not correct question. Pause and work through each option using your knowledge of the backbone chemistry of both lipid classes. Ask, what structural feature does each class have? And what do they share? The statement that is not correct is option C. The reasoning requires knowledge of the backbone chemistry of both classes. Sphingomyelin is built on a sphingosine backbone, which is an amino alcohol. The fatty acid is attached to the amino group of sphingosine via an amide bond, which is a carbon-nitrogen bond. So sphingomyelin contains a CN bond. Now consider phosphoglycerides. They are built on a glycerol backbone, but their head groups, such as choline, ethanolamine, and serine, all contain amino groups, and therefore all contain carbon-nitrogen bonds. So both classes of phospholipids can contain carbon-nitrogen bonds. Option C claims only one class can, which is factually incorrect. Now verify the other options are correct. Option A is correct. Sphingomyelin uses an amide bond for its fatty acid linkage, while phosphoglycerides use ester bonds. Option B is correct. Both classes are hydrophilic at their phosphate-linked head groups and hydrophobic in their fatty acid tails. Option D is correct. Both phosphatidylcholine and sphingomyelin can have choline as their head group. Framework for NOT correct questions. Identify what is structurally shared between both classes, and look for the option that incorrectly claims that a feature is unique to just one class. Question 4. The mechanism by which statins lower LDL cholesterol involves. Option A. Inhibiting HMG-CoA reductase, leading to decreased hepatic cholesterol synthesis, which in turn triggers upregulation of LDL receptors. Option B. Activating bile acid synthesis. Option C. Blocking intestinal cholesterol absorption. Option D. Increasing LDL receptor degradation. Pause. This question tests cascade reasoning. You must trace a cause and effect chain from the drug's molecular target all the way to the physiological outcome. Identify each step before choosing. The correct answer is option A. Statins work through a three-step cascade that requires precise mechanistic reasoning to trace. Step 1. Statins competitively inhibit HMG-CoA reductase. the rate-limiting enzyme of the mevalonate pathway. This pathway is the primary route of cholesterol biosynthesis in liver cells. Step 2. With HMG-CoA reductase inhibited, hepatic cholesterol synthesis drops significantly. The liver's intracellular cholesterol pool is depleted. Step 3. The depleted cholesterol pool activates SREBP, sterol regulatory element binding protein. This transcription factor senses low intracellular sterol levels and responds by upregulating the transcription of the LDL receptor gene on hepatocyte surfaces. More LDL receptors means faster clearance of LDL particles from the bloodstream, and plasma LDL falls. Option B is wrong. Statins do not directly activate bile acid synthesis. That is a downstream consequence, not the primary mechanism. Option C describes the mechanism of ezetimibe, a completely different drug class that acts at the intestinal brush border. Option D is the opposite of what happens. LDL receptor expression increases, not decreases. Framework. For pharmacology questions, always trace the full cascade from drug target to enzyme inhibition to intracellular signal to gene expression to physiological outcome. Never stop at step 1. Question 5. The formation of a phosphodiester bond during DNA replication is driven by Option A. Hydrolysis of pyrophosphate released from the incoming deoxynucleotide triphosphate. Option B. Reduction of NAD+. Option C. GTP hydrolysis. Option D. ATP hydrolysis directly. This question is about thermodynamic coupling, the strategy cells use to drive an otherwise reversible biosynthetic reaction to completion. Pause and think. What is the thermodynamic role of pyrophosphate in this reaction? Why does its removal matter? The correct answer is option A. This is a classic example of thermodynamic coupling. The mechanism works as follows. DNA polymerase catalyzes the attack of the 3' hydroxyl group of the growing DNA chain on the alpha phosphate of the incoming deoxynucleoside triphosphate. This nucleotide L transfer forms a new phosphodiester bond and releases a pyrophosphate molecule. 2 phosphates join together. If the reaction stopped here, it would be reversible. The equilibrium does not strongly favor synthesis. The critical step is what happens next. Inorganic pyrophosphatase immediately cleaves the released pyrophosphate into 2 separate inorganic phosphate ions. This hydrolysis reaction is highly exergonic, approximately -7 kilocalories per mole. By removing the pyrophosphate product from the system, pyrophosphatase drives the equilibrium of the polymerization reaction strongly in the forward direction, making DNA synthesis essentially irreversible in vivo. Options B, C, and D are all incorrect. NAD+ reduction, GTP hydrolysis, and direct ATP hydrolysis are not involved in driving this specific chemical step. Framework. When a biosynthetic reaction is thermodynamically marginal, look for the coupled exergonic step that removes the leaving group and pulls synthesis forward. This pyrophosphate coupling principle applies broadly in biochemistry to nucleic acid synthesis, amino acid activation in protein synthesis, and fatty acid activation by acyl-CoA synthetase. Let us summarize the 5 reasoning frameworks from this video. 1. Chaperones prevent aggregation by shielding exposed hydrophobic patches. The driving force is the hydrophobic effect, not a structural template. 2. Enzymes lower activation energy without changing the equilibrium constant. Always separate kinetics from thermodynamics. And remember that option B is always the classic trap. 3. For NOT correct lipid questions. Check what structural features are shared across both classes before claiming uniqueness. Carbon-nitrogen bonds exist in both sphingomyelin and phosphoglycerides. 4. Statin action requires tracing 3 steps: enzyme inhibition, cholesterol depletion, and LDL receptor upregulation. Never stop the cascade at step 1. 5. PPI hydrolysis by pyrophosphatase provides the thermodynamic pull for DNA synthesis. Coupling an irreversible step to pull equilibrium forward is a principle that applies throughout biochemistry. These are not just answers to these 5 questions. They are templates you can apply to any biomolecules question you encounter. The next video in this series covers methods in biology, the third highest priority unit with over 4,000 questions in our database. Practice these concepts now on GEC JRF Academy at gekjrfacademy.com.